you did in lab, and Chapter 21, which you're doing now on the immune system. As I said, the format of the exam will consist of multiple choice and matching questions and some short answers. It'll be 50 plus questions.
I don't suppose we can have the test sooner than Thursday.
Say again?
Sooner than Thursday before we start going into new stuff.
You should have it on Thursday?
No, before Thursday.
That would be before Thursday? You want it earlier?
I get confused when we start on something new and we haven't tested on the old.
Well, I want everybody to have a chance to make sure they've gone over it. That's why I'm pushing it back a little bit.
Okay.
But we will start the new material. Okay, this will give you the weekend and a couple days to recover
Yeah.
if you wish. Okay, so Thursday, 1 week from today, but yes, we will start new material, which will not be on the exam. Okay? So make a note of that. If you want it earlier,
Do you have the,
we can do it.
like, do you have the papers for the next sections and such as that?
I don't have them yet, but I will have them.
Okay, I was wondering if I might could get them before we start it, and then maybe I could get them
Probably
posted.
on Monday.
Okay.
All right.
If you want to take the test early, can you take the test early?
Pardon me?
If you want to take the test early, can you take the
No,
test?
everybody takes the same time.
I'm not sure.
Probably won't wrap the whole chapter up until Monday. Okay? This will give you a couple days. I mean, if you want it on Wednesday, I can do the whole class on Wednesday. Well, think about it. But anyway. In the meantime, we need to finish the chapter we're on now, which is chapter 21. And as I said, in chapter 17, the toughest part is on the blood clotting. Make sure you know the procoagulants, and the clotting factors, in other words. Make sure you know the anticoagulants and how they work. We went through those. Make sure you know the stages in hemostasis, vascular spasm, platelet plug formation, and then the blood clotting process itself. And in the blood clotting, remember, there are three major phases. The first is the most complicated because that involves a series of reactions. And this is accomplished either by what's called the extrinsic or the intrinsic pathway. The end result is the same, but the way to get there differs a little bit because different clotting factors may be involved. And what starts it is different. In the extrinsic pathway, you've got something released by damaged tissue because extrinsic pathway involves damage to cells or tissues outside the blood vessel, and that's frequently the case. And extrinsic, again, means extrinsic to the blood vessels, not extrinsic to the body. What starts the extrinsic pathway? Tissue factor, or tissue thromboplastid, which is leaked out of the damaged cells. And it goes through a series of chemical reactions. And remember, I'm not going to require you to know the individual clotting factors, which are usually listed in Roman numerals. But I do want you to know that calcium ions are involved, and the end product, of course, is what?
Blood clot?
Prothrombin activator, or prothrombinase. That enzyme then catalyzes the second phase, which is a signal reaction, conversion of prothrombin to thrombin. And the end result of that is another enzyme, thrombin, and that catalyzes the third and final phase, which again is a signal reaction, and that is the conversion of fibrinogen to fibrin. That's the end product of the blood clotting process. The fibrin is a water-soluble protein. That's what makes up the thinning work of Bedford and clot. Within that, of course, you have fat blood cells, like red blood cells, platelets, and so on. Okay? So you should know that. And what starts the intrinsic pathway, which is the other pathway by which you form prothrombin activator? Platelet factor 3, something released by activated platelets. And this is why everything that you need for clotting the blood is already in the blood. So we say intrinsic pathway, it's not intrinsic to the body, it's intrinsic to the blood. So therefore I ask, blood clotting occurs outside the bodies by what pathway you would say, or should say? It's the intrinsic pathway. That's why when you handle blood outside the body, put in a container, it's got to be treated with an anticoagulant or the blood will likely clot, or glass, for example. Okay? That's why tubes, for example, are heparinized. It's an anticoagulant. Okay? So make sure you know that. And then once that clot is formed, of course, you want to get more compact. Involved in that, of course, would be the platelets releasing chemicals. That helps to make it more compact. And then the platelets themselves go beyond that by literally pulling on the thriving threads. And like
Pulling
squeezing a
it together.
sponge, squeezing the fluid, the serum, right out of the clot, making a nice compact mass to shut off blood flow. Okay? And then, of course, there are things in the platelet. There's an inactive enzyme. You should know what that is. And then once the tissue has been repaired, the blood vessel, that enzyme becomes activated, and it breaks down the fiber. What's the enzyme?
Prothrombinase?
Profibrinolysin becomes profibrin, okay? Or fibrinolysin, excuse me. Or plasminogen becomes plasmin, same thing. Okay, and it's activated. It's already in the clot, but it's activated by the platelets. Okay?
So what's the enzyme again?
It's what?
What is the enzyme that causes it?
Called plasminogen becomes plasmin, or profibrinolysin becomes fibrinolysin. Because it lyses the fiber. Okay, I know the chemicals are also involved in repair, okay? Those actually from your platelets, things released by the platelets, okay, and also then by the endothelial cells. All right? I like to say, study that, learn that, and over the weekend, have some time and get this together. Okay, in the meantime, we've been talking about the immune system, and the immune system in the immune system, we talk about innate immunity. And innate immunity is what you're born with. And that basically is non-specific, which means it's directed against anything that is considered foreign by the body. And how does the body know something's foreign? It's because the cell markers, or the chemicals on the cell membrane, or viral particle, or whatever, are different than your own. You have your own markers. Those are called antigens. Those are called self or autoantigens. Your immune system sort of categorizes these or catalogs these around the time of birth. Anything that's encountered after that then is regarded as foreign, and the immune system will attempt to destroy with a response. So again, non-specific means it's directed against anything that is not part of the body. It's not normal. Different antigens. We talk about the lines of defense. What's the first line of defense?
Skin.
The barriers,
Barriers.
the skin and mucous membranes and chemicals associated. What this does is to help things from, prevent things from getting in the body in the first place and failing that, reducing the numbers. Skin's a very good barrier, very few things can penetrate. I mentioned a couple parasites that can, but for the most part, when you have an invasion or infection through the skin, it's because you have breaks in the skin. And again, they can be very tiny or minute because we're dealing with very tiny organisms, microorganisms, bacteria, viruses, and so on. And same thing with mucous membranes. Although mucous membranes, more things can penetrate mucous membranes in skin because the mucous membranes have living cells right to the surface, unlike the skin, which consists of dead cells and stratum corneum. Second line of defense are not barriers per se, but they're cells and various processes that are associated with them. What kind of cells? Second line of defense.
Phagogen.
Phagocytes, okay, these are cells that ingest and digest particulate matter.
Phagocytic.
These would include what kind of cells? Neutrophils, what else? What are the big eaters?
Monocytes.
Macrophages, which are
Macrophages.
monocytes, okay? And also eosinophils. These are phagocytic cells. The eosinophils, though, tend to be targeting the multicellular parasites. The macrophages and the neutrophils tend to target smaller things, bacteria, viruses, and so on. Okay? Also part of that second line of defense is another type of cell that is not phagocytic. What is that? It's a specialized lymphocyte, large lymphocyte. It's called a natural killer cell or natural killer lymphocyte. And those are the large lymphocytes. They're not as common as the common lymphocyte, but like other lymphocytes, they're not phagocytic. They kill by releasing toxic chemicals. So they're called natural killer cells or natural killer lymphocytes. Okay, that's part of the second line of defense. But unlike the typical lymphocyte, which is very specific, these are not.
They're not picky.
They're not specific. In other words, they will target anything that is regarded as foreign. Okay? And how does the immune system know that a cell is foreign? Well, it may be from another individual, another organism, for example, or it could be a body cell that has become parasitized by a virus or intracellular bacterium or something like that. Because when these things get into cells, what they do is they put antigens on the surface of cells. And the immune system recognizes these as being different than the normal. So they will attack these cells. And that includes abnormal cells like cancer cells too. Okay? We've also got a process we mentioned that's localized, part of the second line of defense. What is that? When you get a cut in your finger, okay? It bleeds for a while, stops bleeding, then what happens? Okay, it swells, it gets darker, the temperature rises, and it hurts, okay? What do we call that? Inflammation, okay? Inflammation. And that's usually localized. What's the purpose? Well, first of all, what are the signs and symptoms of inflammation? What's characterized by it? Okay, a darkening or reddening of the skin. What else?
Hot.
Hot, it becomes hotter, okay, than normal. Becomes swollen, enlarges, and usually
It hurts.
okay? And why is this? Because the blood vessels, the small arteries feeding blood to that area, dilate. They bring in more blood. The capillaries that are fed by those small arteries become more permeable, leakier, and they leak more fluid into the tissue spaces. So with bringing in more blood from the core, that elevates the temperature, that also darkens or reddens the skin, and then the fact you've got swelling, of course, that's what causes, is because of that fluid accumulation, that also puts pressure on the pain receptors, so it's usually painful as well. What's the purpose of inflammation? Why does it occur?
To make you slow down and let it heal. Make you stop.
Well, what do you want to do first of all? What's the body want to do? What's to contain the invader, okay? You localize it, keep it from spreading. So you try to wall it off, and then bring in immune cells and antibodies to fight the infection. So it's to localize and keep it from spreading. Not only the pathogen, the disease-causing organism, but also any toxic products it produces. To destroy it, neutralize it, okay? Keep it from spreading. And then also to repair the tissue, okay, as well. There's another process we mentioned, which is fever. You can read that. It's not working. Fever is a body-wide or systemic process. What's the purpose of fever? Why do we often have a fever associated with infection? Well, first off, you got to change the thermostat, right? You're going to change the temperature in the house. What are you going to do? You're going to adjust the thermostat. What is the thermostat in the house? It's the hypothalamus. Hypothalamus is part of the diencephalon. You may remember, does a number of things, but one of the things it also does is to maintain or to set the thermostat of the body. Okay? So how does that thermostat get adjusted? The bacteria can go up and turn up the heat. It's chemicals, right? Cells communicate with chemicals. So what's released? Prostaglandins, okay? These are hormone-like substances, and this causes the hypothalamus to reset or turn the thermostat up. What brings the release of that hypothalamus? Hypothalamus is responding to what? By a compound called interleukin-1, released by phagocytic cells, by white blood cells. And what are they responding to? They just do it randomly? Toxins. Okay, so the bacterium gets in there, organism gets in there, releases toxins, that stimulates the white blood cells to release interleukin-1, and that stimulates the hypothalamus to release prostaglandins, that turns the thermostat up. How does the body temperature increase? Now you reset the thermostat, what happens? You bring the temperature up. You get vasoconstriction, that doesn't really bring the temperature up, but it keeps from losing or retaining the heat, okay? Because you're reducing blood flow through the skin, and the skin is like a radiator, okay? How does the temperature actually go up? You increase the metabolic rate of cells. When cells are metabolizing more actively, what do they need? They need energy. Where does the energy come from? ATP. To produce ATP, they do what? They break down organic molecules, and when they do so, they trap some energy ATP, but a lot of it as heat. They get a little more heat. Okay? And what else? What
Shivering. Shivering.
Okay? You get this chill. And that's muscle contraction. That requires energy from ATP, and again, when you form that ATP, you're also forming heat. That's what brings the temperature up. Okay, and that'll usually be maintained until you basically stop the prostaglandin synthesis or block it. Okay, and that's what fever relievers usually do, like aspirin, acetaminophen, acetaminophen and stuff like this, okay? Now, the fever can be really useful because the interleukin-1 that's released by the white blood cells, you can read it, but it stimulates our lymphocytes, both our B and T lymphocyte production. So it increases lymphocyte production to help fight the infection. Also, it basically intensifies the effect of an antimicrobial compound, which we'll talk about in just a minute, called interferon. It's called interferon because it interferes with viral replication. It can also sequester, kind of hide away some important minerals that the bacteria may need for reproduction, like iron, zinc, and some of these other things. Then also, it basically accelerates tissue repair because it basically increases metabolic rate. So a low grade or moderate fever is useful. So it's not always a good idea to knock the fever out right away. It's when it gets too high that can be a problem, because then it can cause denaturation of proteins. And our enzymes and our reactions in our body are catalyzed by enzymes. Remember, these are molecules that speed up chemical reactions. Enzymes are mostly proteins, and proteins generally are very susceptible to extremes of temperature. High temperature will denature them. What do we mean by that? It changes three-dimensional shape or configuration. Why does that affect an enzyme? It's protein. You change its shape.
It won't fit into the phone.
You may remember enzymes work with the substrate, or compounds they work on, by forming an enzyme substrate complex. They have on their surface what's called an active site. That fits the substrate. That's how they catalyze reaction. If you change the shape of the molecule, you change the shape of active site, doesn't fit the substrate, and it doesn't catalyze reaction. So reaction will go without an enzyme, but so slowly as to be useless to the organism. I mean, we can digest our food without digestive enzymes, but it might take you weeks to do so, by which time you'd have starved to death before you get any nutrient value from the food. That's why we have digestive enzymes, to speed up the process. Okay? So low-grade fever, moderate fever is good. If it gets too high, then there's a danger. It can cause denaturation of proteins and shut down a lot of very important reactions in cells, particularly very metabolically active tissues. And a prime target, of course, would be what? The nervous system. That's why high fevers, prolonged, can cause severe and even permanent brain damage. Okay. All right, also part of the second line of defense are some antimicrobial chemicals. And one of these, and it's more than one actually, is complement. Complement is actually a series of proteins. And they react in a certain sequence of reactions called a complement cascade. You don't need to worry about the name of it. But the end result is something very important. What complement does, once it's activated, it lyses cells. It breaks up cells. And we call that cytolysis. Cyto refers to cell, lysis means to break it up or break it apart. So it basically punches holes in the cell membrane. Yes?
Can you enlarge this?
I can't. That's the problem. The thing is jammed, okay? It's not working. That's why I said, did anybody have a hammer? Really heard that. I didn't say that. Sometimes you love to. Okay? But anyway, cytolysis or cytolysis, lysing or breaking up the cell membrane. What it actually does, and we're not going to go into great detail. If you take microbiology, we'll cover it in a little more detail. It actually punches little holes, nice neat little pores, called membrane attack complexes. I call them little MACs. Okay, not big MACs, but little MACs. That's what they are. Membrane attack complex. But they're nice, neat little pores in the cell membrane. That causes cytoplasmic contents to leak out, and you destroy the cell. That's the end result of complement. It lyses cells. It also enhances inflammation, okay, because it causes cells like mast cells, basophils, and platelets to release these chemicals, these inflammatory chemicals like histamine, for example. And this brings in more phagocytes. And then another process, opsonization. It will literally coat a cell. Remember I said when cells get coated with protein, these can be antibody or complement, they usually readily get phagocytized and destroyed. Okay, we call that opsonization. So again, complement works by lysing cells. It enhances inflammation by stimulating the release of inflammatory chemicals like basophils, mast cells, and so on. Brings in platelets. And then it can coat cells, which makes them more vulnerable to phagocytosis and destruction. And people have problems with complement, this can be inherited, okay? Often much more susceptible to diseases than those people who have normal complement. Okay? So complement, antimicrobial proteins. And normally they just circulate in the blood, inactive, but once they get activated, then these results. And the main thing that you see is lysine cells. It breaks them apart. All right, something else is interferon, another antimicrobial chemical. And interferon is so called because it interferes with viral replication. Now, viruses are a little bit different. Viruses are not cellular, unlike bacteria. They're much smaller than even bacteria, which themselves are very tiny. In fact, bacteria have their own viruses. They're called bacteriophages, their own parasites. And they get into bacterial cells and destroy bacterial cells like this virus is coming to our cells, destroying our cells. But what viruses are, basically, are a packet of nucleic acid, either DNA or RNA, not both. Cells have both RNA and DNA, as you know. Both bacterial cells are our own cells. Viruses have one or the other, but not both. So we have a class of viruses called DNA viruses, where they carry the genes in the DNA, which is what we're used to. And another group of viruses called RNA viruses that carry the genes in the RNA. They have no DNA. But again, the virus is a packet of RNA or DNA surrounded by a protein coat. And then some viruses go beyond and have what's called an envelope, which is a modified cell membrane of the cell in which they are replicating in the body. And because cells don't have a metabolism of their own, they don't have any organelles, they have to get into a living cell in order to replicate or reproduce themselves. What interferon does is prevent that replication in cells. So it's called interferon. It is produced by cells once they get infected with a virus. But it does not protect the cell that is infected. What it does, it is released by that cell, it's picked up by adjacent cells that are not infected, and stimulates them to produce antiviral proteins. To make a long story short, what it does in these cells is to shut down the protein synthesis machinery in that cell. And since the virus is dependent on the cell machinery to replicate, that is to reproduce its nucleic acid and its protein from the protein coat, which protects that nucleic acid, it shuts down viral replication. Now this is not, it shuts down all replication of protein, which means the cell can't do it either, but it's a temporary thing. And what this does is give the cell time to recover, expel the virus, without actually causing it harm. I mean, if it persisted, then the cell would die too, because it didn't make the proteins. But it's long enough to basically stymie the virus, keep it from producing proteins, and then eventually it clears the virus, and then it can make its own proteins again. So it's a temporary cessation or shutdown, excuse me, of protein synthesis in the cell. That's what the interferon does. Again, it doesn't protect the cell that's infected, just enabling cells that are not yet infected by shutting down protein synthesis temporarily in those cells. And the interesting thing is, it is host cell specific. A virus can stimulate the production of interferon. It can protect against other viruses too. But interferon only works in our own cells. Only human interferon works in human cells. Other animals produce interferon, dogs, cats, and so on, but it only works in those species, not in our own. So it's species specific, but not virus specific. Okay? We have different kinds, major types of human interferons, alpha, beta, and gamma. And basically, the alpha and beta are very similar, but they're produced by different cell types. Alpha interferon is produced by white blood cells other than lymphocytes. Beta interferon is produced by fibroblasts. Remember, these are cells in our connective tissue that produce the protein fibers in our connective tissue. And gamma interferon is produced by sensitized lymphocytes. And alpha and beta interferon are also called type 1. They basically inhibit cell growth, they're shutting down the protein synthesis, and they also can suppress tumor formation, which is why sometimes interferons may be used in cancer treatment.
Isn't it real painful for people now?
Pardon me?
Some people, it's real painful.
Say again?
Painful to be treated with interferon.
Sometimes you can treat interferon certain types of cancers, but it does
Yeah.
interfere with protein synthesis. And protein or cancer cells are actively producing protons because proteins rather, because they're dividing. So sometimes can target these cells.
But is it really painful?
Is it what?
Painful? That's,
Pain?
yeah, she was asking if it was
I said
painful.
Very painful.
Very painful.
To be treated with that.
I don't know. Because I'm not familiar with the treatment, but I don't think it'd be any more painful than what they use with cytotoxic drugs. Because it can kill cells, and the radiation therapy, which also kills cells. Because those will kill some normal cells too. That can be very painful. Also, it affects the appetite, and of course, hair production goes out just when the hair falls off, and you lose the appetite, and they have trouble. Yeah, chemo, and radiation therapy can do it too. That's why people that have these treatments just look terrible. Because they're losing weight, they lose their appetite, they often can't keep food down, so it's very difficult to maintain normal body functions.
Chemotherapy, is that a chemical therapy?
Yes.
Because that chemo
It's cytotoxic,
taxis,
it's cytotoxic chemicals that are killing cells. And what you're trying to do is target the actively growing tumor cells, but you will in fact kill some normal body cells as well. And same thing with radiation therapy, you're trying to target on the tumor. But you're going to have skin kill some cells around it as well.
Is that why when you have a child, they don't, if it's doctor, they don't give them like any medicine? Because the interferons are usually successful with stopping the protein synthesis enough to kill the dog?
So that's the purpose. What's your question?
She says that way they don't
I
give
say, is that why they
medicine
don't
for a
prescribe any
virus.
medicine? Because usually
for babies.
for babies.
Oh, yeah. No, you wouldn't. Because you've got actively growing cells there in the babies, and of course, a lot of protein synthesis. So, yeah, a lot of these drugs you can't use on infants, they're very, very young.
You need antibiotics, because the virus isn't alive.
Well,
I know, but I'm just.
antibiotics don't work on viruses.
I know not antibiotics, but they don't give you anything.
Yeah, they won't give them babies, yeah.
Liver.
Okay, what they used to do to get interferon was they would harvest it from cadavers.
Oh,
And only get
wow.
a little bit at a time, okay? So the expensive process and also potentially dangerous process, because anytime you transfer something from one body to another, there's always risk of infection, particularly with viruses. What they do now, though, is they can basically insert human genes into the genome of microorganisms like bacteria and certain yeasts, and they will then basically translate those genes, and they will produce human interferon, which they can do. And that's a safe way to do it. You can put it in Escherichia coli, for example, or Saccharomyces, which is yeast, insert those human genes, and then they will basically
Multiply.
transcribe those genes, and they will produce those human products. And they've done that for clotting factors, for example, for hemophiliacs, and also for
Wow.
interferons. And you'll probably see more and more of that. Okay? That's called recombinant human interferon. The recombinant DNA basically is when you put DNA from one organism into another. And then these organisms, microorganisms, can use them to make things that are very helpful to us, like interferon, for example. Okay, any questions on any of this? Okay, this ends our discussion on the innate immunity, which is nonspecific. I'd like to turn your attention now to the adaptive immunity, which is specific. And now we're talking about a response directly against a specific agent, a particular bacterium, a particular virus, a particular fungus, whatever. We call this specific or adaptive. This is what you acquire during your lifetime. Innate is what you're born with. Adaptive is what you acquire as you come in contact with foreign antigens. on foreign cells or viral particles. There are several characteristics here of specific immunity. And first off is, as the name suggests, it's specific. It's directed against a particular agent, a particular pathogen, a particular cell, a particular viral particle. Very specific. Also, it's body-wide. It's systemic. And something very important, it has a memory. It remembers foreign antigens that you've encountered before. So you build up an immunity. Once you've been exposed to a parasite, and you get this from having a disease and recovering from the disease, or with an immunization. That's the whole basis for immunization. You can be exposed to the parasite in a form where it's not going to cause a disease. It can either kill or inactivate, what we call attenuated. That will stimulate the immune response without having to go through the ravages of the disease. That's why we have immunizations. A lot of people will get the immunization when it's far worse or far less severe than having a disease. Okay? And that's because we have cells in the immune system that are so-called memory cells, because that's what they do. They remember the antigen. And next time you encounter it, you respond much more quickly and much more efficiently with the response. The result is you usually don't get sick, or it's going to be much milder than the original. Okay, so again, specific response is specific, like it's a specific agent. It is systemic, it's body-wide, and it's got a memory. It remembers antigens, foreign antigens that you've encountered before. Okay? Now there are different types of immunity. I'd like you to know this. When we talk about active immunity, this means immunity resulting from your body's actions. It's something your body is actively engaged in. That's called active immunity. As opposed to passive immunity, where you're getting protection from another source, usually another person. That's passive immunity. So active immunity, your body is actively engaged. Passive immunity is protection from another source. So we have four types here. Naturally acquired active immunity. This is what you get when you get a disease and you recover from it. You're left with some protection, having had the disease. It's naturally acquired active immunity. It's active because your body has responded by producing an immune response, which allows you basically to fight off the pathogen and recover from the disease. And sometimes the protection is lifelong. Measles, for example, or yellow fever, just to name a couple. You get the disease, you don't get it again. Naturally acquired active immunity. Naturally acquired passive immunity is something we've all got as babies. The mother will pass antibodies across the placenta into the fetal circulation. And for the most part, these are protective antibodies. So when that baby is born, it is protected against diseases to which it's never been exposed, but the mother has. And the mother has some protection in the form of antibodies. Those antibodies cross the placenta, get in the fetal circulation, and this is passive, because again, the protection came not from the baby, but from the baby's mother. And that's why babies, are protected right off. They have some protection. If it weren't for that, most babies would never survive. Right away, as soon as they're born, they start coming in contact with foreign organisms, some of which are pathogens. It could be fatal, causing fatal infection. Okay? If the mother breastfeeds the infant, even better, because there are also antibodies in the breast milk. And she extends that passive immunity as long as she is breastfeeding, especially during that first year. That's why it's encouraged mothers to breastfeed. I know it's inconvenient, okay? So you gotta say, well, breastfeed the baby. You didn't have to do it. There are antibodies in the breast milk. So generally speaking, breastfed babies are much healthier than bottle-fed or formula-fed babies. It's especially important in these developing countries where a lot of people don't have access to good medical attention. We used to send vast quantities of formula over these countries, which we're actually doing them a disservice, because the problem was not that the formula wasn't nutritious, but the the mothers often had to mix it in dirty water. And what they were doing basically was feeding their infants this formula mixed with contaminated water. They were basically infecting their infants with a lot of potential diseases. So it basically helped to increase infant mortality as a result. It's much better if the mother breastfed the baby because, again, she'd be passing antibodies in breast milk to her child. Okay, but again, that is naturally acquired passivity. It's acquired naturally, but it's passive because, again, the protection, usually in the form of antibodies, come from the mother. Those antibodies have crossed the placenta into the fetal circulation. And again, if the mother breastfeeds, the antibodies also in the breast milk will extend that passive immunity. Okay? And especially the initial secretion, that's called the colostrum irritation antibody. And usually the breast milk starts to flow shortly after birth. The problem with passive immunity is, unlike active immunity, passive immunity is relatively short-lived. So it may only last for a period of a few months. Active immunity, of course, produced by the body, is much longer duration. It's like I say, sometimes it's lifelong. Okay, we also have what's called artificially acquired immunity, and we can have artificially acquired active immunity. This is what you get when you have an immunization. Artificially acquired active immunity. You're vaccinated or immunized with an organism that can cause disease, but that organism either been killed or it's been attenuated, weakened, so it can't cause disease. But what it does is to stimulate the immune system to respond with an immune response. You build up antibodies and immune cells against that organism. And a common vaccine that most people have, and they get it as infants, is the polio. The original polio vaccine was a kill vaccine. That was a so-called Salk vaccine. And it came out about the mid-50s. I think they had the first clinical trials about 1955. And they ran extensive trials in this country, and it was very effective. Polio was a disease that everybody knew about because, not that it was such a great killer, any more than other childhood diseases, but it primarily affected children. And some of those children were left paralyzed, permanently paralyzed. The disease was often called infantile paralysis. And way back when, it's way before your time, they had something called the March of Dimes, okay? And kids would bring in dimes, literally, to school. The schools would collect it, and they would send it in to the government, literally to the White House. And they just got hordes of mail with dimes. And those dimes were basically put into polio research. And that helped to contribute to the formation of a vaccine. And like I say, the first vaccine was the Salk vaccine. That was an injected vaccine, and it was killed. Okay? Problem was, there was one lab, I think it was out in California, they didn't treat it properly, they didn't kill the virus, and they were injecting live virus. So that actually brought about some case of polio. So for a moment, they had a little black eye, but they quickly corrected that, but the vaccine was very effective, they found it. The problem was, though, it had to be injected, okay? And it was more expensive. In the meantime, the Sabin vaccine eventually, a couple years later, replaced it, and that was the one that was taken orally. And by the way, Salk and Sabin didn't like one another. They competed with one another. In fact, Sabin, the older man, was constantly trashing Salk. You know, he said, "Well, that vaccine you can make in the kitchen chemistry. You know, it's not worth anything. The good vaccine is the live polio vaccine, which I'm working on, which later came out." And for a while, that replaced the Salk vaccine, because you could get it orally, like a little sugar cube. They would have been. Maybe you got that. But what has happened eventually is that they found that the Sabin vaccine, the oral one, sometimes mutates and sometimes can become viable and produce polio. And two, people that have that oral vaccine, since it's a live virus, they shed the virus, and if it comes in contact with people who are not protected, they can get polio. So there's a downside to that. So now I think most of the vaccine now is back to the old Salk vaccine. If you've had your kids immunized with a polio, was it not the injected one? Or was it this oral one? Okay. I think now they're doing more of the Salk vaccine than the oral one, at least at this time. They found that the disadvantage of the oral one is you shed live polio virus, and you can spread it to other people. But I think in most foreign countries, particularly those in developing countries, the Sabin vaccine is cheaper, and it's easy to administer. All you have to do is administer it orally. You don't have to inject it. So it's cheaper and it's easier to administer, and that's why a lot of countries still use the oral vaccine.
Okay, is that the one that most older people have the scar with?
No, that's for smallpox,
Smallpox.
which you no longer get. Yeah, you can tell the age of a person how is that they got a smallpox, okay? Yeah. We stopped giving smallpox vaccinations, I think, late 60s, early 70s, something like that. And yeah, it does leave a scar, because what they do is they basically abrade the skin. I've had one, so I know what it's like. All right?
Me
In fact, I
too.
gave somebody one when I was in school, graduate student. But anyway, yeah, you got one.
Yeah, I got one.
You had a question, somebody? Right.
That scar, you also get it if you get on the TB vaccine, the BCG.
No.
No.
Oh.
I don't think so with BCG. Has anybody had a BCG? Because they don't routinely give that in this country, BCG. You've had one?
Yeah, it leaves a scar, so you get it.
It leaves a scar?
Yeah.
Is it like a,
Yeah.
is the scar like smallpox? Okay. They give in the UK? They still give them? Yeah.
Yeah, well, I got the smallpox when I was little, and that scar hasn't faded any. It
It
stayed
never
there
does. It
forever.
never does. It's still there. Yeah, it'll be there. But, okay, so you do get a scar from the BCG. I don't know, I'm not familiar with that, because we've not given the BCG in this country. BCG is for tuberculosis. And it was given especially to children because it was thought to basically help control and prevent tuberculosis in children. But it was never really accepted by the FDA here in this country, so it's not been given. But if you've had the BCG, you will test positive for the skin test for TB the rest of your life. So if you have that, in your nursing you'll have to have that, a skin test. You better tell them you've had a BCG. Most of you have not, but you would.
Yeah, I got it, but I still, the skin test, it doesn't rise. I'm fine.
You don't get a
but I
positive
have to
skin
do the
test?
chest
I
test.
thought they said you don't have to do the skin test anymore, do you?
Well, I still have to do it, but then if it's gonna rise, so if it rises, you have to do the chest X-ray.
But you never
to
test
me-
positive? You don't test positive? Do you test positive? You haven't had one?
I've never had one. So
Never had one? Okay. Usually you do test positive. Probably did a lousy job. I don't know. Smallpox, yes. They don't give smallpox anymore because they figured, World Health Organization decided that you were more likely to have a reaction from the vaccine than actually acquire smallpox. Smallpox is so rare, they think it only exists now in some research laboratories in the US and in Russia.
Wow.
But the danger is it'd be an ideal terror weapon because we haven't been exposed to smallpox for years. And smallpox, untreated, can have a mortality rate of about 30%. And it can be deadly. And it's very contagious. So it's potentially very dangerous stuff. But like I say, we don't give it anymore. I think some people get it, like some troops and stuff like this will get it, but it's unusual. They don't give it to the general public. Like I say, if you have that, you'll have a scar, okay, where they give the smallpox. What they do is they braid the skin and inject, and in doing so, they put the virus in. Like I say, I've had one, and when I was in school, took a course in medical microbiology when I was working on my master's, we gave one another smallpox vaccinations. Mine did not take, which means my life partner gave me one, I gave him one. Mine didn't take, which means I was still immune. His, he got a real reaction. He was not immune. So, or maybe I did a better job. I don't know. But we don't do that anymore, okay? So it'd be rare to find somebody who's had a smallpox vaccination, unless they're elderly like me, okay?
And me.
But I probably wouldn't be any better protected than you by now because it's been so long.
So that's not one of the lifetime ones?
No, it's not lifetime. It's not lifetime. So hopefully we won't have these smallpox epidemics because that could be very deadly if it ever broke out. Okay, anyway, these are artificially acquired active immunity. You introduced the organism into the body and you stimulate the immune response, just like you have the disease, but you don't have to go through the ravages of the disease. Okay? And that's called a vaccine. Okay, and it can be killed, the organism can be killed, it can be attenuated, where it's been rendered weakened, so it doesn't cause the disease, or in some cases, you don't even inject the organism. It may be a chemical it produces, like the tetanus vaccination. You're not actually injecting the organism, you're injecting what's called a toxoid, which is the inactive form of the tetanus toxin. So you'll produce antibodies, antitoxins, so should you come in contact with the tetanus organism and that neurotoxin, you neutralize it. You don't get the disease, which is nice. You don't want to get tetanus. Okay, and lastly, artificially acquired passive immunity. This is something you get, we're injected usually with a solution containing antibodies against the disease organism. And usually this is given in an emergent situation. You've been bitten by a rabid animal, okay? And you don't want to get rabies, because rabies usually fail to most people. And usually what they do, if you've been bitten by the neighborhood dog or cat or something, okay, they confine it, they hold it and see if it develops any symptoms of rabies. If it does, then you go through the regimen. If not, you're off the hook. Or what if you're bitten by a wild animal and it runs off in the brush? You feel lucky? Yeah, that thing wasn't rabid, you know. And then suddenly you come, because once you come down to symptoms, usually too late. The only good thing that be said about rabies is it has a long incubation period. But once you start getting the neurological symptoms, usually it's fatal. So it's a good idea to get the regimen. And people who are handing animals, for example, probably a good idea, like working in vet clinic, to have that vaccine. And two, keep in mind too, that if you're picking up animals along the side of the road, you know, kind-hearted dudes, the strays, you know where that animal's been. And it could be carrying some of the disease like rabies or distemper or whatever. So,
But it only lasts a few weeks,
what?
that vaccine.
Yeah, because it's passive. It's passive. But what they do, when you get the rabies shots, they'll give you the shots, they're injecting the antibody to protect you, and they'll also often give you an injection to get some active immunity too. But you want to neutralize the thing before it has a chance to bring about the signs and symptoms. Because what the virus does, it affects the nervous system and eventually migrates down the salivary glands. And it affects the animal to make it more aggressive, so it's more likely to bite. When it does, of course, the saliva contains the virus, and you basically are injecting the saliva into the individual. And that's why rabid animals often act rather bizarre and often very aggressive. Okay, so you've got an animal that's normally afraid of humans, and it comes up to you and it's that kind of strange, beware. Okay, like I say, don't worry about the squirrels around here, because you've been feeding those practically, so that's different. And you can always see that they're foaming at the mouth or something like that.
So it's like a snake bite, but then you
Yeah, whatever. Okay. Any questions on this? Okay. So all these different kinds of immunity. All right. Types of immune response that we mentioned, these all involve T lymphocytes and B lymphocytes. First off, the humoral immunity. This is also called antibody mediated. Humor refers to body fluid, and this is where the antibodies are found, not just in the blood, but also in other body fluids as well. So we call this humoral or antibody-mediated immunity. And that's because it involves antibodies that are produced by sensitized B lymphocytes. And these antibodies, again, are in our body fluids. The cellular immunity, or cell-mediated immunity, involves T lymphocytes. And these are cells, basically, have nothing to do with antibody production. Instead, we have T lymphocytes that will attack and kill abnormal cells. Parasites outside the cells can be attacked and destroyed by antibody, but once they get inside cells, they're protected. This is where the T lymphocytes come in, those so-called cytotoxic or killer T cells, because they will destroy these cells, break these cells open, and then the antibody can get them. So it basically is a way to attack those intracellular parasites, viruses, and bacteria that have gotten into cells. It can also be directed against the protozoa, fungi, helminths, and so on. But it's a double-edged sword, because our T lymphocytes, which will destroy these abnormal cells and destroy these intracellular parasites, can also turn on cells in a tissue transplant or an organ transplant. It's usually the T lymphocytes that result in graft rejection, because they recognize the antigens on those tissues in that organ are different than your own. If you don't have something to basically suppress the immune response, you often outrightly reject the organ. Unless you're lucky enough to have an identical twin, and then you've got the same basically antigens. But most of us don't have that luxury. We have to get the organ from another person. Hopefully one closely related by blood, because you're more likely to have more antigens in common and fewer foreign antigens. There's always a risk of rejection. That's why when you get an organ transplant, such as an identical twin, you're going to be on immunosuppressive drugs probably the rest of your life. You knock down the immune system. It's a fine line. If you knock down too much, you could die from a common infection. If you destroy your immune system, suppress it so much it doesn't work. And not enough, you may just outright reject that organ because you're too lymphocyte.